cryo-EM. It's a method to obtain three-dimensional structures of molecules. The main challenge is the images are very, very noisy, like a TV set that is not functioning very well. You can think of every individual image in cryo-EM basically as a piece of the puzzle. We can have million pieces of the puzzle, we have million images. Trying to solve this puzzle becomes very, very challenging. In my group, we do the development of computational tools and algorithms. And so to get to high resolution, from the computational point of view, we need to process those very large data sets. It's rather a very complicated algorithm to integrate and analyze all the information in the images to predict the 3D structure. The product, at the end of the day, that we have is a software toolbox that structural biologists can then use in their own labs and analyze their own data set that they acquire using their electron microscopes. The long-term goal is to enable biological researchers to better understand the 3D structure of the proteins and the other molecules, and eventually perhaps even be able to cure diseases that have to do with those proteins.